Scientist monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host and moderator for today's webinar. Today, Dr. Lisa Monteggia will present mechanisms of antidepressant effects. The Brain and Behavior Research Foundation funds research around the world that identifies the causes and improves treatments that will ultimately result in preventative techniques and cures for mental illness. Since 1987, the Foundation has awarded more than $328 million in research grants. We are the largest funder of mental health grants outside the federal government. 100% of all donor contributions for research are invested in the grants given directly to our researchers who are working to find scientific breakthroughs in disorders such as ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, OCD, PTSD, and schizophrenia. I'm delighted to introduce Dr. Lisa Monteggia. Dr. Monteggia is the Ginny and John Ulick Professor at the University of Texas Southwestern Medical Center at Dallas. She is a 4-time NARSAD grantee and the recipient of the Foundation's 2005 Friedman Prize awarded to a young investigator for exceptional basic research. Her lab focuses on the role of the molecular and cellular basis of neuroplasticity as it pertains to neuropsychiatric disorders. Her research encompasses molecular, cellular, behavioral, and electrophysiological approaches using mouse models. Today's webinar will begin with Dr. Monteggia's presentation. This will be followed by Q&A. To submit a question, please use the Questions tab on the control panel of your screen. Feel free to submit any questions throughout the presentation. Following the presentation, I will present your questions to Dr. Monteggia, and she will address as many of them as time allows. Now I'm very, very pleased to present Dr. Lisa Monteggia. Lisa, the floor is yours.
Thank you, and thank you for that kind introduction. I'm very delighted to speak about some of our work today on rapid antidepressant responses. As Dr. Borenstein said, NARSAD really has been instrumental in our research. It was really the first grant I received as a postdoctoral fellow that really spurred my lab to focus on antidepressant responses and to try and understand them. So I'm going to talk to you a little bit about what we've done over the past 10 years, really trying to understand mechanisms of antidepressant action. So with that, so to start with, what is depression? And I put this slide up here because we all know that depression is a serious mental condition, something that we hear about. People talk about it all the time in many different ways in terms of, I feel depressed, I feel blue, I feel down. But it's a psychiatric disorder that's characterized by clear symptoms that are outlined here. Symptoms of anxiety, anhedonia, loss of appetite. Many different aspects make up depression. And it's because of this sort of characterization of symptoms that makes it very difficult to try and understand what really drives depression. What's the underlying pathophysiology? Because it's such a wide range of symptomology that can occur, and patients can present very differently. But there's not only major depressive disorder, there's dysrhythmic disorder, minor depression, things like psychotic depression, postpartum depression. These things can all have very different effects in terms of how they manifest and how people talk about them. So it's sort of a term that's used in our culture in many different ways. But the reality of it is major depression is the leading cause of disability in the U.S. And I find that rather staggering when you think about that, when we think about all the different diseases and how we hear about prevalence, cause of disability, and all of that. What I have on the bottom right is a graph. And while this graph is a few years old, I like this graph a great deal because it actually shows over the last several years of period between 2005 and 2008, the 12-month prevalence of depression among U.S. adults by sex. And on the left is females and on the right is males. And you can see that females have a range right around 9%, and males right around 5% in terms of the 12-month prevalence, with an overall U.S. population of around 6.7%. Now, what's interesting is that, as you'll notice, females have about a twofold high percent increase in prevalence compared to males. However, it's really unclear why that is. Females, some people have suggested, may be more susceptible because of estrogen. Some people have suggested that males may be more resilient because of testosterone effects. And some people have argued that it really is not a sex hormone-specific event. So it's really unclear. Current treatment, primary treatment, is through serotonin selective reuptake inhibitors. They're the most common prescribed drug and treatment for depression, but its mechanism of action is really unclear, and that's what we're going to talk about. Typically, there's this time lag where it takes several weeks to reach clinical efficacy, but not all patients actually do reach clinical efficacy. There's actually a large percent that don't, and those patients with time are the ones that are most at risk for suicide. And I put the numbers up here because I found the numbers to be actually rather surprising. In 2003, the number of suicides in the U.S. was 31,484, and the number of homicides was around 16,000. In 2013, the numbers for suicides jumped to over 41,000, while the number of homicides stayed roughly equivalent. It's really sort of staggering, the increase in suicides we've seen over the last 10 years, but yet it's not a topic that we feel comfortable talking about. We really understand very little about the causes of suicide. As I mentioned, SSRIs are commonly used for the treatment of depression, but they're by no means the only treatment. Psychotherapy is also used often in combination with antidepressants, and electroconvulsive therapy and other brain stimulation therapies are also used, with electroconvulsive therapy very widely used for patients that haven't responded to other treatments, very safe and very effective. Our focus initially, in terms of entering the field of antidepressants, was based on observations that a growth factor called BDNF, brain-derived neurotrophic factor, appeared to be increased by various antidepressant treatments, in particular with different drug treatments, as well as with electroconvulsive therapy, electroconvulsive shock in animals. So we asked the question, is BDNF required for an antidepressant response? And we used mice that we had generated in which the growth factor BDNF had been selectively deleted in the brain. And the question was, would those animals still respond to an antidepressant? What we found in the data shown in the graph, this is data from the forced swim test, but there were multiple paradigms that were done with this. What we do is we take an animal, a mouse in this case, and we put it in a beaker of water. And very quickly, the animal, as they're swimming, they learn to sort of give up. They just float to keep their head above water. When you give an antidepressants, the antidepressants actually cause a decrease in immobility, and that's suggested to be an antidepressant response. The currently approved antidepressants all produce this sort of decrease in immobility. So this test is commonly used to measure sort of antidepressant-like responses, if you will. What we found was that just deleting BDNF had no effect on this sort of behavioral effect, if you will. If we gave a low dose of daciperone, as it's commonly used, what we found was a significant decrease in immobility, suggestive of an antidepressant response in littermate control animals. However, that effect was not observed in the BDNF requirement, showing that BDNF appeared to be required for the antidepressant effect. And as I said, we did this with multiple paradigms and with multiple antidepressants, and we consistently saw that the loss of BDNF impaired the ability for the animal to respond into the antidepressant. We followed those studies up by actually trying to identify where in the brain BDNF seems to be mediating this effect. And we started off first with different lines of conditional knockout mice. And the one thing that they seemed to all have in common was the deletion in the hippocampus. So we decided to focus in and ask, is BDNF in the hippocampus required for this antidepressant response? So the hippocampus, which is shown here, is a large area. And what we did is we used a viral mediated approach to actually delete BDNF in either the CA1 region, which is shown here, or in the dentate gyrus, which is shown here. And what we found is that when we deleted BDNF selectively in the CA1 region, this top region here is shown, that the control animals, where they just had a virus expressing just a green fluorescent protein without any effect, that the antidepressant still worked, that the antidepressant worked as expected. You see the significant decrease in immobility. In the animals that had the CA1 deletion, mediated by Cre recombinase, you can see again that desipramine produced an antidepressant response, suggesting that BDNF in the CA1 region was not required. However, when we looked in the dentate gyrus deletions, what we found is that again in the control animals that had AAV GFP, those animals as expected responded to desipramine. However, that did not occur in the animals with the localized knockout in the dentate gyrus, suggesting that BDNF in the dentate gyrus was required. And we went ahead and looked at other behavioral paradigms and other antidepressants. So this data really suggested that BDNF played a critical role with antidepressant responses. And we've really spent the last several years trying to understand how do you go from sort of increasing monoaminergic transmission, the sort of increase in serotonin and norepinephrine and dopamine to some extent that occur with traditional antidepressants, to mediate this requirement for BDNF. And as we've been doing this, we were actually, as many in the field were, very surprised about the findings regarding ketamine. And ketamine is an NMDA receptor antagonist. It actually blocks a glutamatergic receptor, the NMDA receptor, and it's been around for a long time. Very high doses of ketamine can produce anesthesia. More mid-level doses can produce sort of psychotic, psychomimetic effects. But what was done was that at an extremely, extremely low dose, 0.5 milligrams per kilogram of ketamine, this dose was infused, a single acute infusion, into treatment-resistant major depressed patients, and they actually had a rapid antidepressant response. And in some patients, it actually went out for several days, even out to a week or two. And this initial study was published in 2000, and since then, it actually has been replicated several times in small clinical trials. This just shows you from one of those papers from Zarati and all from 2012, just sort of the rapid antidepressant effect, where the ketamine infusion produces this sort of rapid effect within 30 minutes or so. Out 2 hours, you can see the effect. And in these patients, it was back by 7 days, sort of compared to placebo. So we were particularly intrigued by this finding. And what really inspired us on this next set of research I want to talk about was really the idea that we've been struggling with this, how do typical antidepressants work? How do you get an antidepressant response? Yes, for traditional antidepressants, you need BDNF, but again, antidepressants take several weeks to work. So how do you go from that several weeks of drug administration to actually produce an effect? There's many, many, many papers out there on different pathways, different genes, different proteins that are upregulated. But what are causative for this effect? And that's been very difficult to understand. How do you link it to a particular receptor to mediate the effect? With ketamine, it was a single infusion of a drug that blocks an NMDA receptor that produces a behavioral effect. So to us, the excitement really was we could actually think about a cellular mechanism to mediate the effect. How could you block the NMDA receptor and link it to a specific intracellular signaling cascade to mediate the effect? So the first thing we did was we actually took ketamine and we did a dose-response curve to really focus in on the low dose of ketamine to show that it could actually mediate an antidepressant effect in rodents. And in our studies, we're primarily using C57 black 6 mice. And what we could find is that a single acute injection of the drug, and this is the forced swim test again. Most of the data I'm going to present to you is the forced swim test, even though we've done multiple paradigms. Just for the sake of time, we're going to focus on this. And what we found again was that the single injection of ketamine within 30 minutes could produce a significant decrease in immobility, suggestive of an antidepressant response. And we found that this single acute dose could produce a long-term effect 6 days later, again suggestive of an antidepressant effect. We then took this low dose, and this low dose was selected because it didn't have any effects on learning and memory, on locomotor activity, or producing any sort of disruption in sort of schizophrenia-like behavioral models. So we took this low dose and we tested it in several different behavioral paradigms that are supposed to test antidepressant responses, if you will. I looked at the tail suspension test, learned helplessness, sucrose consumption, novelty-suppressed feeding, social defeat, a whole range of those. And what we wanted to do was to see, would ketamine produce an antidepressant effect, this low dose, in these paradigms? And it seems sort of paradoxical because these paradigms predict antidepressant effects. Of course, ketamine should work, but all the antidepressants that are currently available all target monoamines. So it's always been unclear that if you have an antidepressant that works via a different mechanism than monoamines, would it actually work in these paradigms? And we found that low-dose ketamine worked in most, but not all of these paradigms. So we decided to focus on the paradigms that worked, including, as I've shown you, the forced swim test. One thing what we did is we took animals, and to sort of model depression, if you will, we stressed the animals. We did what's commonly called chronic unpredictable stress, where we do 2 mild stressors a day uncontrollably for 21 days. We then gave a low single injection dose of ketamine, and what we found was that in 3 different paradigms that I'm showing you here, over on the left, in terms of percent sucrose intake, ketamine actually triggered an increase in sucrose intake suggestive of an antidepressant effect. In the forced swim test, you see a significant decrease in immobility, again suggestive of an antidepressant effect. And in the novelty-suppressed feeding, again, as you see a decrease in latency to feed, suggestive of an antidepressant effect. So it appears this low dose of ketamine actually does reverse sort of behavioral phenotypes associated with stress in rodents. So with that, we decided to ask our first question. And the question was, is ketamine actually producing its antidepressant effect by blocking the NMDA receptor? That's ketamine's known target, but it could potentially have off-target effects. So to answer this, what we did is we took, again, our low dose of ketamine, or CPP, which is a competitive NMDA receptor antagonist, or MK801, which is a use-dependent NMDA receptor blocker. We did dose-response curves on CPP and MK801 to identify low doses that, again, don't have the confounds of hyperactivity or learning and memory deficits can be associated with NMDA receptor antagonists. And what we did is we took either ketamine, CPP, or MK801 and injected into a cohort of mice, and 30 minutes later examined their behavior. And we did this at either 30 minutes or in different cohorts 3 hours, different cohorts 24 hours, or in still different cohorts 1 week. We wanted to make sure that at each of these data points there was no animals that had been retested. We wanted to avoid the confounds of behavioral habituation. What we saw with ketamine was that, again, this is the forced swim test data, that low-dose ketamine produces a significant decrease in immobility suggestive of an antidepressant effect within 30 minutes, and that those effects persist out for a week. CPP also produced a very rapid decrease in immobility suggestive of an antidepressant effect. This effect persisted out for 24 hours but was gone by 1 week. And MK801 had a very dramatic effect in terms of a significant decrease in immobility at 3 hours, but by 24 hours the effect was gone. So this data suggested to us that blocking the NMDA receptor appears to be necessary for triggering an antidepressant effect. However, what was intriguing to us is that ketamine, CPP, and MK801 all have half-lives in the very short range, approximately 2 to 3 hours. So therefore, these long-term antidepressant effects are likely not due to persistent blockade of NMDA receptors, but likely to some type of synaptic plasticity process. So with that, ketamine blocks the NMDA receptor and produces an antidepressant effect. So how do we think about mechanism? Well, as I told you in the initial slides, we had previously identified BDNF as a critical component for typical antidepressant responses. So we ask, is this molecule also involved with the antidepressant effects of ketamine? So we took our BDNF mice, which are shown here again in black. The littermate controls are shown in white. Again, this is the forced swim test data, and just deleting BDNF has no effect on antidepressant responses, as you can see. If we give the low dose of ketamine, ketamine triggers a significant decrease in immobility, suggestive of an antidepressant response, but that effect is not observed in the BDNF knockouts. And we see this both 30 minutes and 24 hours later following ketamine. We also confirmed this with MK801, the other NMDA receptor antagonist that was shown in the previous graph. MK801, again, in the littermate controls, produces a significant decrease in immobility suggestive of an antidepressant effect. That doesn't occur in the BDNF knockouts. And if we look 24 hours out later, Ketamine does not produce an antidepressant MK801, does not produce an antidepressant response at this time point in agreement with our previous data. So it appears that we block the NMDA receptor and that downstream of that, BDNF is required for the antidepressant effect. So how is BDNF linked to this process? Well, we looked first to see if this BDNF component really was involved in terms of TrkB signaling, because BDNF is linked to its high affinity TrkB receptor in terms of mediating downstream signaling. So we first wanted to further confirm this BDNF effect. So we generated conditional TrkB knockout animals, and just deleting TrkB, as you can see, has no effect on this behavior. Low-dose ketamine produces a significant decrease in immobility suggestive of an antidepressant effect that's blocked in the TrkB knockouts. And we characterized these animals through a whole range of paradigms. And again, ketamine did not produce an antidepressant response in any of the paradigms we looked at with the TrkB knockout. If we just took C57 animals and gave them a low dose of either ketamine or MK801, and looked at TrkB autophosphorylation, sort of a sign of its engagement, if you will, we dissected out the hippocampus 30 minutes after ketamine or MK801 administration, and we saw an increase in phosphorylation of TrkB as shown here, no change in total TrkB, suggesting that BDNF TrkB were engaged in the antidepressant effects of ketamine. So we then said, okay, can we in some way link blockade of NMDA receptors to BDNF? So what we initially did was we took a low dose of ketamine or MK801 and we administered it to C57 animals, and we sacrificed them either 30 minutes or 24 hours later. We then dissected out the hippocampus and looked at BDNF mRNA expression as well as BDNF protein expression. And what we found was that, rather surprisingly, ketamine or MK801 at either 30 minutes or 24 hours after administration did not impact BDNF mRNA expression as determined by qPCR to the coding exon of BDNF. However, rather surprisingly, ketamine or MK801 both produced a significant increase in BDNF protein within 30 minutes following the drug administration that was back down by normal at 24 hours. And this was rather surprising to us because this is a rather robust change in BDNF protein at 30 minutes following administration. We looked at this is by Western blot analysis. If we looked on the blots at the pro-BDNF bands, it followed the same pattern, suggesting that this could potentially be an increase in BDNF protein that was occurring. We also, though, confirmed this by ELISAs, just another way to look at BDNF and protein, and we saw the same effect, that BDNF protein was rapidly upregulated following ketamine administration, and it wasn't sustained. And the reason why we really focused on this is because previous work had shown that several years ago, had shown that if you just infused recombinant BDNF protein into the hippocampus of mice, that you trigger an antidepressant effect even days later, 6 days later after this recombinant protein has been put in. And what's intriguing about that is that the BDNF protein is going to be degraded pretty quickly, but yet it's still producing an antidepressant effect 6 days later. So how is that? Is BDNF mediating some sort of plasticity that's occurring in terms of the antidepressant effects? So we first sort of came back to this question of BDNF. Is this really an increase in BDNF protein due to translation? So to do this experiment, what we did is we inhibited protein translation with a drug called anisomycin. And these are just blocker experiments where we simply administered either our drug anisomycin and then ketamine 30 minutes later, and then checked on behavior either 30 minutes after that or in a separate cohort of animals 24 hours later. And what we found is just administering anisomycin has no effect on, again, this forced swim test behavior, that a low dose of ketamine in C57 animals, again, produces this rapid antidepressant effect, but if anisomycin is present, you block the antidepressant effect. And we saw this both at 30 minutes as well as 24 hours, demonstrating that protein translation is important for the antidepressant effect. We also took animals and just looked at BDNF protein at these points, and as you can see, just with ketamine by itself, again, it confirms our previous results. BDNF protein is significantly upregulated in the hippocampus within 30 minutes, and anisomycin with ketamine blocks the effect. It's just correlative data, but it goes along with our BDNF data. We went ahead, in experiments I'm not going to show you, we went ahead and looked at if we inhibited transcription. Even though BDNF mRNA expression was not altered, other genes could be altered that could be necessary. So we did the same type of blocker experiments shown here using actinomycin D. And what we found was that ketamine in the presence of actinomycin D still produced an antidepressant effect. So at least this rapid, this early 30-minute time point of antidepressant effect wasn't requiring new gene expression. It was just protein expression that appeared to be driving the effect. So the question is, how do you go from blocking NMDA receptors to upregulating BDNF protein expression, triggering downstream effects? And that actually was a question we had a hard time really wrapping our heads around, because there's a lot of data out there where you can link NMDA receptors to changes in protein expression and downstream synaptic plasticity processes such as LTP. But those processes all require activation of the NMDA receptor. And here it appeared to be blockade of the NMDA receptor. So we decided to look, is there any way that activation of the NMDA receptors could also be producing this antidepressant effect? And so what we did is we gave low subepileptic doses of NMDA and tested the animals in various behavioral paradigms for antidepressant responses. And as shown here, there was no change in immobility, demonstrating that low-dose NMDA is not producing any sort of rapid antidepressant response. And what's shown here on the right is just to show that there was the expected increase in ARC protein, showing that the NMDA did have effects on molecular targets within the brain, even though it wasn't mediating the behavioral effect. We looked at several different possibilities to try and explain how we go from blocking NMDA receptors to upregulating protein expression to mediate the antidepressant effect. One theory that we looked at very closely was this disinhibition idea. And the disinhibition idea involves the idea that ketamine, or NMDA receptor blockers per se, actually preferentially block NMDA receptors on inhibitory neurons, thereby raising overall levels of excitation, and it's through activation of NMDA and AMPA receptors that you can mediate the effects. So we decided to look at this as well. And as I showed you, it didn't appear that activation of NMDA receptors was triggering our effect. So we went ahead and looked at AMPA receptors. So we took and blocked AMPA receptors with a drug called MBQX, and it also did not produce any antidepressant-like effect. So I said, okay, perhaps it's not activating NMDA or AMPA, perhaps it's overall excitation. So we tried to just raise overall excitation by giving a drug called picrotoxin, and again, we could not trigger a rapid antidepressant response. What's shown here in the middle and on the right is just that these drugs do have the expected effects. Picrotoxin does produce this effect on molecular target of ARC, even though it doesn't produce this rapid antidepressant effect. And I should add that after this data was published, there have been two studies out, one of them in which NMDA receptors were deleted selectively on paraalbumin inhibitory neurons, and it was shown that ketamine still produces an antidepressant effect in those animals. A second study has come out showing that when NMDA receptors, NR2B in particular, are deleted from excitatory neurons, you lose the antidepressant effect of ketamine. So that data is very much supportive of what we're seeing, that the effect appears to not be due to the sort of disinhibition idea in triggering the antidepressant effect per se. So while that's not due to it, what actually is triggering the antidepressant effect? Well, as we went down this path to try and understand this, we did an experiment that actually turned out to be actually quite important for our studies. As I've shown you, a single low dose of ketamine produces this rapid antidepressant effect as shown here. Blocking AMPA receptors doesn't trigger a rapid antidepressant effect. However, ketamine plus MBQX actually prevents the antidepressant effect. And MBQX again blocks AMPA receptors. So just blocking AMPA receptors doesn't trigger the antidepressant effect, but it's clearly very early involved. And what we were able to do through data I'm not going to show you is that it appears that ketamine, through upregulating BDNF protein, this BDNF protein is triggering the insertion of GLUR1 and GLUR2 that are required for this antidepressant effect. We did this work through doing experiments called biotinylation experiments to actually show there was an increase in both GLUR1 and GLUR2 on the cell surface in the hippocampus of neurons. So we said this insertion of GLUR1 and GLUR2 are often linked to plasticity. Is there any type of plasticity that's involved? So what we did is we took a hippocampal slice and we simply added ketamine and looked at sort of what happened. And typically when you cut the slice and you add an NMDA receptor blocker, there's not much that happens per se, but you add the drug for a very short amount of time. But the antidepressant effects of ketamine aren't observed just at a short amount of time, a couple minutes. It takes about 30 minutes before the antidepressant effects are observed. So we added ketamine for 30 minutes, and as we recorded, we actually saw a very, very nice potentiation that's observed. We're quite intrigued about this, of how do you block an NMDA receptor and produce this potentiation. And we can show in experiments I'm going to show you in a minute that these are BDNF dependent. But what we ended up coming back to after a lot of work was the following pathway and the following idea that we explored for our mechanism of antidepressant action. So synaptic transmission in the brain sort of comes in two flavors, if you will, the sort of evoked transmission or just the background spontaneous transmission. And over the last 7 to 10 years, there's been quite a bit of interest in this role of spontaneous transmission and evoked transmission. And what's shown here, this is a neuron, and these are the vesicles that contain glutamate. What's been shown is that these pools that contain glutamate appear to be different if you release, if an action potential comes, if it's sort of evoked transmission versus spontaneous transmission, the sort of fusion that just occurs in the background. So these pools appear to be different. Work has shown, using the use-dependent effects of MK801 to look at the NMDA receptor, that when glutamate actually gets released by evoked transmission, it actually causes an increase of calcium coming through the NMDA pore, and you activate an enzyme called Kinase 2. But for spontaneous transmission, it appears that when glutamate is released, you activate a different pool of NMDA receptors. And there's no data at this point to suggest it's due to different subunit composition. The idea has been more in terms of spatial temporal regulation of where it is located in the synapse based on some modeling work. So in this instance, with spontaneous transmission, glutamate is released, binds to the receptor, calcium flows in again through the NMDA receptor, but instead of activating Kinase 2, you just activate a different signaling pathway called Kinase 3 or eukaryotic elongation factor 2. And what's interesting about this kinase is that it only is known to have one substrate. It phosphorylates this one enzyme, eukaryotic elongation factor 2. And when this kinase phosphorylates this enzyme, what happens is you have an effect on protein translation. So when phosphorylation event occurs, you basically upregulate protein expression. Work has shown in primary hippocampal cultures that if you give a low dose of an NMDA receptor blocker and block spontaneous transmission, you can inhibit this kinase such that the phosphorylation event doesn't occur, and you can rapidly upregulate protein synthesis. And what's been shown has been an upregulation of GLUR1 and GLUR2. So what we did is we actually went back and did a lot of work repeating previous studies to actually show that ketamine could block spontaneous NMDA receptors and inhibit this kinase in vitro and actually try to show the selectivity of it. And we were able to do that. So with that, we actually went in vivo and asked, is this the pathway that mediates ketamine's antidepressant response? So we took the low dose of ketamine or MK801, administered a single low dose to C57 animals, and what we could see was that ketamine and MK801 both produce a significant decrease in phosphorylation, and that's just shown by this decrease in brightness, if you will, in the hippocampus. And we quantitated this. If we take C57 animals, give them the low dose of ketamine or MK801, sacrifice them 30 minutes later, we see a decrease in phosphorylation of EEF2, sort of our proxy that the kinase has been inactivated, if you will. So it appears that this pathway is actually engaged in vivo. So we block the NMDA receptor, we inhibit this kinase such that we are upregulating protein synthesis. So we ask, if we're correct with this idea, then if we just inhibit this kinase, independent of blocking NMDA receptors, can we trigger a rapid antidepressant response? So to that end, we took two drugs, and these are EF2 kinase inhibitors, Ritalin, which is shown on the left, and NH125. And I should mention that these drugs, while they inhibit EF2 kinase, they also can have other effects on various kinases in the cell. But for the purposes of this, we gave the low doses of Ritalin or NH125 after we'd done dose-response curves. And we showed that if we just administered these drugs, within 30 minutes, we get an upregulation of BDNF protein synthesis in the hippocampus and a decrease in phosphorylation of EEF2, which you would expect if it was actually inhibiting the kinase. Behaviorally, what happens is that these drugs produce a significant decrease in immobility, suggestive of an antidepressant response within 30 minutes that is present 24 hours later and extends out to a week in the case of Ritalin. So it appears that independent blocking of the NMDA receptors, we can trigger a rapid antidepressant effect. And if we take this inhibitor and put it in the BDNF knockouts, which we know BDNF is required for the ketamine effect, it's also required for this Ritalin effect. So what we see again is that these are our BDNF knockouts. These are the littermate controls. If we give the low dose of Ritalin, similar to what was seen in C57 animals, we get a rapid decrease in immobility, suggestive of an antidepressant effect, but not in the BDNF knockouts. So this data really sort of suggested to us that this low dose of ketamine, which blocks spontaneous NMDA receptors, is working through the inhibition of EEF2 kinase, upregulating protein synthesis. A number of the proteins that we see appear to be dendritic protein synthesis because it's not just BDNF, but there's other proteins that we've observed as well. And these dendritic proteins can actually impact synaptic plasticity. So with that, we've sort of tried to follow this up in many different ways. And the one thing is, as I mentioned, the pharmacological inhibitors with EEF2 kinase. While those drugs do inhibit the kinase, they could have off-target effects. So we've been able to obtain the EF2 kinase knockout animals. We've characterized those through a range of behaviors. And what we see, not surprisingly, is that these, without the kinase, you don't have phosphorylation of EF2. If we take these animals, this EF2 kinase knockout animals or littermate controls, and give them a low dose of ketamine, that the wild-type littermate controls have this rapid increase in BDNF protein, but you don't see it in the EF2 kinase knockouts, demonstrating that EF2 kinase is upstream of the BDNF effect. If we take these EF2 kinase knockout animals and we look at behavior, I'm going to show you here again the forced swim test or the novelty suppressed feeding test, that just knocking out EF2 kinase doesn't have any effect on this behavior. A low dose of ketamine produces a significant decrease in immobility suggestive of an antidepressant response, but this effect is lost in the EF2 kinase knockouts. So this is the most direct data we have that EF2 is required for the antidepressant effect of ketamine. We saw similar results when we looked at the novelty suppressed feeding paradigm, where again in wild type animals, ketamine produces a significant decrease in the latency to eat, where that we don't observe that in the knockout animals. Again, demonstrating the requirement of EEF2 kinase for this behavioral response. So EEF2 kinase is required. It appears through its inhibition that you upregulate BDNF protein. And as I mentioned, this BDNF protein appears to trigger this insertion of AMPA receptors and this sort of plasticity. So we ask, is this plasticity also dependent on EEF2 kinase? So we went back and we took our hippocampal slices, and again, we added ketamine for 30 minutes, washed it out, and we see this nice potentiation in the littermate controls. But what we found in the EF2 kinase knockout animals is that this potentiation was blocked, demonstrating that EF2 kinase was required for this potentiation. We saw the same effect with BDNF. BDNF was required for the behavioral effect. If we took our BDNF knockouts, again, cut a hippocampal slice and added ketamine for 30 minutes, washed it out in the littermate controls, we see this nice potentiation that's lost in the BDNF knockouts. This potentiation that we see is kind of surprising, again, because it's elicited by a block of NMDA receptors. We went ahead and showed that if we add ketamine in the presence of anisomycin, this protein translation inhibitor, which blocks the behavioral effect of ketamine as an antidepressant, it also blocks this potentiation. And we see it both with ketamine and with MK801. And one of the last pieces of data I want to show you, we actually focused in on this idea that we were blocking spontaneous NMDA receptors. We're not blocking all receptors everywhere. What we're arguing is that we're blocking spontaneous evoked, but spontaneous are actually weighing in to mediate this behavioral effect. So what we did is, in looking at this potentiation, if we added ketamine with TTX, which blocks evoked transmission, you can see that ketamine still is triggering this potentiation, which argues for the segregation or this distinction between evoked and spontaneous. Moreover, if we just add TTX by itself to the hippocampal slice, we don't induce a potentiation. If anything, we see a decrease. So this sort of adds to this idea that the potentiation that we see that's correlated nicely with our behavior appears to be due to spontaneous transmission. So our current model that we have is, again, this is our synapse, is that with low-dose ketamine, what's happening is that we're blocking NMDA receptors, and we're blocking in particular spontaneous NMDA receptors. We block these receptors, we inhibit EEF2 kinase, such that the phosphorylation event doesn't occur. We upregulate protein translation, not only BDNF, but like I said, other proteins as well that we've identified. We focused on BDNF because of our knockout data, and we're introducing this potentiation. So we're doing a lot right now, trying to look at sort of downstream effects of what is this potentiation? What is it good for? It correlates with the behavior, but can we more conclusively link it to the behavior? We're continuing our work on BDNF and TrkB, really trying to understand their roles in this process. We're also trying to look more closely at EEF2 kinase. What is it doing? Where is it located? How is it regulating this BDNF effects? And we're also trying to more closely look at spontaneous transmission. Are there ways to manipulate spontaneous transmission that could perhaps trigger a rapid antidepressant effect independent of blocking NMDA receptors and potentially the side effects associated with blocking NMDA receptors? So as we work towards the future, I hope what I've shown you today is that we're focusing, as many people in the field are, on this faster acting antidepressants. There's a clear unmet public health need for more rapidly acting antidepressants. Ketamine, while it has potential side effects, it provides an important proof of principle approach to understand the mechanism of action. Ketamine actually shows us that it's possible to generate a rapid antidepressant response, and also by the fact that it's linked to the NMDA receptors allows us to actually think mechanistically about what is required. We can actually move past just the fact of giving a drug for weeks on end and seeing effects and not understanding causality to actually, based on the acute nature of the treatment with ketamine, the single infusion, to actually think mechanistically, how is this working? And can this actually contribute towards the development of new therapeutic targets? Another point, though, is that an important aspect of this is actually being able to identify who would respond to ketamine. And ketamine may be able to provide a way to identify biomarkers for fast-acting antidepressant responses. It's really a big unmet need in the field in terms of biomarkers for depression as well as for most psychiatric illnesses. So I presented to you our work on preclinical animal models and how we're trying to elucidate antidepressant responses. We've been able to put forth a novel hypothesis of how ketamine may work, linking it mechanistically all the way to how the drug can actually bind its receptor all the way down. And we're trying to explore this hypothesis and further, test what happens, what are the requirements for this, what does this mean, with the hopes of generating new therapeutic targets. And I think this research is critically important because as we talk about how we're going to develop new approaches, you really have to understand mechanism. how does something work? And by understanding mechanism, really that has been the way the field has moved. And I say field, really the scientific field. Psychiatry has been really much more difficult to tackle, but when we think about the major advances that have been made in many different fields, it's really been because of basic research. If we think about, for example, cancer, back several decades ago, if someone had a diagnosis of cancer, people didn't really discuss it. They didn't really talk about it. In most cases, it was a death sentence. But we've made advances on understanding many, many different types of cancers and treatments. And depending on the type of cancer now, there's actually prognosis. People actually, if they're identified early enough and seek treatment, can have very, very good outcomes. It's not uncommon to hear someone say that they're cancer-free. It's the same with HIV in the 80s. Initially, people didn't talk about HIV because it was a death sentence. There was really nothing you could do. People were shunned. But very quickly, basic research focused in and identified treatments for HIV that have really been dramatic. Quality of life just has been amazing for people with HIV. It was a death sentence to now being able to have a long, normal quality of life. And so we're hoping that as we work towards the future, we can take these types of ideas that are coming from basic research, these hypotheses, and move them towards treatment advance to really provide hope as well as very much needed treatment advance for these patient populations. So with that, I will close and turn the microphone back over to Dr. Borenstein, and I just want to acknowledge the people on the screen that were all responsible for various aspects of this work.
Thank you, Lisa, very much. This is extremely important work, as you said, and I think that it demonstrates the real hope that basic science brings to the table in terms of developing new approaches to treating depression and other psychiatric conditions. So this is extremely important. One of the questions that a number of people asked is, from your perspective, given the very basic work that you're doing to understand the mechanism of action, what would you see as a reasonable timeline to the development of new medications that can act in this rapid, have this rapid effect to treat depression?
I think that it's a very exciting time. The work with ketamine that's been done clinically clearly demonstrates that you can have a rapid antidepressant effect. Because of this, there have been a number of studies where they've tried other NMDA receptor antagonists, and the majority of them have not worked, or they haven't worked as well as ketamine. And that's been an issue. So I think that by understanding the basic mechanism per se, it might allow us to do better targeting, better drug development, if you will, because it doesn't appear it's just blocking the NMDA receptor sufficient. Not all NMDA receptors antagonists do this. So I think we're at an exciting time to actually try and elucidate and really focus in on what is the key requirements in blocking this receptor that trigger the effect. In our hands, we think it's the EF2 kinase. So our hope is that over the next several years, as we further refine these targets with companies having different libraries available, that we might actually be able to set up screens to actually try to facilitate this. And there's a lot of interest going on clinically. There's numerous large numbers of clinical trials going on to further delineate out these different drugs and to try and understand why ketamine works and most of the other ones are not working.
Very good response, very important response. So people can understand, again, that relationship between basic research and actual development of clinical treatment. One of the aspects of ketamine, the rapid-acting antidepressant effect, is also the rapid-acting anti-suicidal effect.
Yes.
And I'm curious what if your work has been able to shed any light on that aspect of ketamine.
so I think yes and no. So we're studying rapid antidepressant responses, and these rapid antidepressant responses are important not only for individuals that suffer from depression. Ketamine has also shown clinical efficacy in patients that are bipolar, as well as a small study where patients were presented at a psychiatric ER with suicidality, and there were beneficial effects observed. So I think our study has clear implications. If there's a rapid antidepressant that actually can move forward, there's many patient populations that will benefit. Definitely people that are suffering from suicide. Based on the initial indication, there's such a need. Hopefully this would be something that would provide relief. Currently there's no treatments, as you know. Patients, if they presented a psych ER, are just admitted. So if they could quickly be stabilized, that by itself is a huge advance. So while our work per se is not really focusing on the pathophysiology of depression or suicide, I view those really as separate questions. We're really trying to focus on this treatment advance. I think the implications clearly are there for suicide as well as other patient populations.
Okay. Well, one of the things that I think is extraordinary, and it goes back to what you said about discoveries about cancer and HIV, is that not too long ago, for instance, when I went to medical school, I was taught that old brains don't grow new cells, and old was after the age of 2. And now we obviously know about neurogenesis, and that old brains, adult brains, and even senior brains can grow new cells. And I'd like you to talk a little bit about your research and how that relates to our greater understanding of the mechanism of neurogenesis.
So neurogenesis has attracted a great deal of interest in the field, and there's clearly been shown that typical antidepressants increase neurogenesis. And the role of neurogenesis in antidepressant responses, though, has been under debate for many, many years. While antidepressants clearly increase it, this debate of how it occurs, is it important in terms of the causative effects or not, have been a hot area of research. In terms of the ketamine responses, our data would suggest, just the clinical data would suggest that neurogenesis is not required for the rapid antidepressant effects of ketamine. It happens too fast. Ketamine is producing antidepressant responses within 30 minutes to 2 hours. So neurogenesis, you clearly can generate an antidepressant response independent of neurogenesis per se. That's, I think, what the ketamine data shows. Now, that doesn't mean that ketamine might not be having some effect on neurogenesis to produce a sort of sustained effect. That's really unclear. So I think that there's still a lot of work to be done on this area. We ourselves, because we've been focusing on this rapid time point, have not investigated neurogenesis.
Okay. But what you are certainly looking at the sort of the synaptic plasticity related to this.
Yeah.
So it's sort of the, increasing connections, development of connections between nerve cells.
Yes, and so we're trying to understand that. Is the changes in neurotransmission, and I think this is why, this is another reason why this work has been really exciting to the field, is that, with ketamine, as we're looking at it, as we're trying to understand behaviorally how it works, there's been so many advances in technology to really look at function. What's happening in terms of synaptic transmission? We're able to actually look at that in ways that, years and years ago wouldn't have been possible. So how is ketamine impacting synaptic transmission? What are the specific effects? And there's so many different ways that you can regulate synaptic transmission, but ultimately it's through those regulations that you're going to trigger behavioral effects. So we're trying to understand the connections, the neurocircuitry that's actually happening to actually mediate the behavioral effects.
Okay. Lisa, I so much appreciate your joining us for this presentation, and even more importantly, the work that you're doing. And just to reiterate really what you were saying before, the potential here is extraordinary, that this is a real game changer in psychiatry. And the point that I think of is clinically, if a person has a severe depression and is at risk of hurting themselves, now they need to be admitted to the hospital for treatment and so that they're safe until the treatment takes effect. But imagine a day when the person could be in an emergency room, get treatment, and go home that day safely, continue treatment on an outpatient basis, medicine, talk therapy, but also go to work the next day or go to school the next day and continue with their life in a full and safe fashion. That's a game changer in psychiatry. So the work that you're doing is bringing us closer to that dream and bringing that dream to a reality. So thank you very, very much.
Well, thank you for having me today and allowing me the opportunity to present some of our work.
Good. Well, thank you. I also want to thank our audience for joining us today. All of the research we fund is made possible through private donations. So if you'd like to make a gift, please visit our website, bbrfoundation.org, or call 1-800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it with a family member or friend, please visit our webinar page at our website. And finally, I hope that you will join us again next month when Dr. Daniel Pine, who's a Foundation Scientific Council member and a NARSAD grantee, and serves as the Chief of Child and Adolescent Research in the Mood and Anxiety Disorders Program at the National Institute of Mental Health, will present a webinar entitled Child and Adolescent Anxiety, Psychopathology and Neuroscience. This will take place on Tuesday, June 9th at 2 o'clock Eastern Time. Once again, thank you for joining us and enjoy the rest of your day. Take care.